The purpose of the bottle biology is to model an ecosystem that demonstrates the relationship between the aquatic world and the terrestrial world around us. In order to create a bottle biology ecosystem model, you will need a 2-liter bottle, gravel, plants, water, soil, wick or cloth to make wick, scissors, and tape. Insects, bugs, and fish are optional. After creating our bottle biology, we predicted that the plants would begin rooting and grow in our bottles because we believed the wick would provide enough water for the plants. Our bottle biology was placed in the windowsill. This is the data we collected for weeks 1 through 6. During our week 1 observation, we noticed that the soil was moist, evidence of condensation, normal H2O levels, and plant growth. During our week 2 observation, we noticed the soil was dry, evidence of condensation, normal H2O levels, and plant growth. During our week 3 observation, we noticed the soil was moist, evidence of condensation, normal H2O levels, little algae growth, plant growth, and little wilting. During our week 4 observation, we noticed the soil was moist, no rooting, condensation, normal H2O levels, little algae growth, plant growth, and wilting. During our week 5 observation, we noticed the soil was dry, little rooting, evidence of condensation, normal H2O levels, algae growth, plant growth, and wilting. During our week 6 observation, we noticed that the soil was moist, little rooting, evidence of condensation, normal H2O levels, algae growth, plant growth, and more wilting. We believe that physical factors such as uncontrollable temperatures and uncontrollable lighting affected the plant's growth. We also believe that possible algae, fungus, mites, or other critters may have possibly affected the plant's growth as well. The darkling beetle experiment is a way in which metamorphosis can be observed. The purpose of this experiment was to observe the life cycle of a tenebrio and to observe their diet. In order to begin a tenebrio observation experiment, you will need mealworms, plain oatmeal, a clean, clear container, and an empty egg carton, and food is optional. First, we got 10 to 12 mealworms and placed them in a clear container with oatmeal. Then we tested the different types of food that we predicted that they would eat. We tested apples, potatoes and boiled peanuts, large potato slices, Rice Krispie cereal, as well as spinach, kale, carrots, celery, grapes, and Frosted Flakes. We also observed the darkling beetle resort to cannibalism. We were also able to observe the life cycle of the tenebrio. The darkling beetle life cycle begins as an egg. After 1 to 4 weeks, emerges a larva, also known as a mealworm, for 8 to 10 days, and molting can occur 10 to 20 times before turning into a pupa for 1 to 3 weeks. Finally, the pupa hatches into a beetle and can live 1 to 3 months. 1 to 2 weeks after hatching, the beetles are ready for mating. A few days after mating, the female burrows and lays eggs. She lays about 300 eggs, and the life cycle repeats. Here you can see younger larvae that have hatched from an egg compared to the older larvae, which are larger. We have collected data for weeks 1 through 6 on the number of alive and dead mealworms and beetles that were present. We currently have 9 mealworms and many babies, 9 alive beetles, and 1 dead beetle. We have concluded that Tenebrio do not eat particular foods because they were too salty or acidic. When they are not fed, they resort to cannibalism because they are scavengers of our environment and they clean up after plants and animals. Tenebrio also prefer dark environments to avoid predators. During our week 1 observation, we noticed black spots on the paper towel. We also noticed the emergence in plant 1 of the primary root and the first leaf. In plant 2, we noticed the hypocotyl, and in plant 3, we noticed the primary root. During our week 2 observation, we noticed an increase in black spots on the paper towel. In plant 1, the second leaf has emerged as well as branching roots. In plant 2, the hypocotyl is still visible. In plant 3, the first and second leaf have emerged. During our week 3 observation, we noticed the paper towel was dry, so we added water. During our week 4 observation, we noticed the paper towel was dried out, so we added more water. The leaves for plant 1 and plant 3 began to turn brown. During our week 5 observation, we noticed a significant increase in black spots. The leaves for plant 1 were green, and the leaves for plant 3 had wilted. During our week 6 observation, we noticed another significant increase in black spots. The leaves of plant 1 began wilting, but the roots of plant 1 and 3 were still white. We have concluded that the corn kernel's growth was stimulated due to the warm temperatures, sun exposure, and absorption of water from the bag. The mold growth was possibly stimulated from the moisture in the bag and the high temperatures while the bag was sealed. In our 6-pack, we have planted 5 deep and 5 shallow sunflower, kale, and safflower seeds. Throughout the course of our observations, we watered our seeds and plants. We only had 4 shallow and 3 deep sunflower seeds sprout. Eventually, 1 deep sunflower sprout died. At the end of our observation, all of our plants began to wilt. We believe the kale and safflower seeds did not sprout because the environment was not favorable due to the environmental conditions.